Hello, and welcome back to Introduction to Genetics and Evolution. In most of the videos that you've seen so far, we were studying patterns of genetic inheritance, looking at alleles from one gene at a time. In the last video you saw, we studied patterns of genetic inheritance, looking at two genes at a time rather than one, but focusing on genes that are on different chromosomes. And we discussed how those assort independently. They follow Mendel's law of independent assortment. In this video, we'll continue to look at patterns of inheritance of two genes at a time, but looking at genes that may actually happen to be on the same chromosome, but maybe not right next to each other. Now, this is called crossing over, and this is a subset of the overall process of recombination. Now, many of you use Wikipedia, I know. Recombination, you can basically break up the word as I've done here. Re for a new, combination. So basically recombination is shuffling of alleles into new combinations, combinations that were not found in the parents. I will emphasize the Wikipedia definition in this regard is not precise. The Wikipedia definition focuses specifically on one type of recombination called crossing over, which will be our primary focus. But I want to emphasize that all recombination is important, both for genetics and evolution, and independent assortment is one type of recombination. Essentially, you have recombination when you produce a gamete that has a combination of alleles that was not found in one of the parents. This gamete is then called recombinant. Okay? So you have to examine this by looking at essentially two stages. You have to look at the person, the sort of focal person. You have to look at the gametes that made this person, things from their mom and their dad, and you have to look at what happens in their gametes to see this. So let me illustrate this with an example. Let's say that you're looking at something on the X chromosome, which is one chromosome, and let's say you're looking at the two traits that I talked about in the last video, being colorblind versus not colorblind. And let's say you're also looking at a trait that's controlled by one gene, and something similar to what I described before with the left thumb landing on top, as opposed to the right thumb landing on top. Remember, the one with the left thumb landing on top was dominant. So if you have a sperm from somebody who is not colorblind and with left thumb on top, so this is the sperm that's coming in. Here's the egg with the X that's colorblind and right thumb on top. You end up with somebody who's heterozygous for both, right? He has one copy of each allele. Now, in this case, this individual who's a female is going to have an egg. Note now this egg will through this process of independent assortment is just as likely to give this X as that X. In this particular case, this X is the one that lands in the egg. And this other chromosome that has the left thumb on top is also the one that lands in the egg. Note this combination is found in this egg, the XCB and the L. The XCB actually came from the mom, from this individual's mom. The L came from this individual's dad. So we actually have a new combination. This is a combination that was not one of the forms that made the person, right? This was not something found in the parental sperm or egg, but it is a new combination. That is why this is called recombinant, right? Because we do not see anybody up here who's XCB and capital L. So what's the opposite of a recombinant? The opposite of a recombinant is a parental type or non-recombinant. So the first part of this figure is exactly the same as the previous one I showed you. But in this case, what happened is this ended up in the egg, and this ended up in the egg. Now, these two are the exact combination that was found in one of the parents, right? So this would be considered a parental or non-recombinant gamete. Do you see what I mean? So basically what you're looking at the gametes that made the individual and the gametes that are coming out of the individual, and you're looking to see, is there a new combination in the gametes coming out of the individual? If there is, then you've had recombination. Now, there's two means of recombination. One of them is independent assortment, which we've talked about, and this is typically what happens when you have different chromosomes, right? That you're just as likely to pass on one copy of the chromosome as another copy of the chromosome based on Mendel's laws. Now, the other possibility is crossing over. Now, again, sometimes the word recombination is synonymized to this. Sometimes people, when they say recombination, are referring specifically to crossing over. In this case, you're actually breaking off a piece of one chromosome and sticking it to another. This right here is a picture from Thomas Hunt Morgan's book from about 1916, where there's a white chromosome and a black chromosome. Pretend that one came from the dad, one came from the mom. And you see in the offspring, you may get this part black, part white chromosome. So it's this, again, new combination, but it's actually all is happening along the length of one particular chromosome. So let me recap what these two things are. First, in terms of independent assortment. Importantly, we can study independent assortment by multiplying probabilities, right? Because we assume that traits are inherited independently. So again, if you're just as likely to pass this gamete or pass on this allele as that allele, we can then just multiply the probabilities to get what's going to happen among all the offspring. So in this example here, this is one of the ones we did in a previous video. If you're heterozygous for both the T and S, this is what would happen in terms of T's. This is what would happen in terms of S's, right? If you're big T, little t, cross to big T, little t, 1/4 of your kids will be big T, big T. 1/2 will be big T, little t. 1/4 will be little t, little t. Same thing is happening with S's. So then to see what would happen for the combinations, we just multiply the two things together. So your probability of being big T, big S would be 1/4 times 1/4, or 1/16. That's what this is right here. All of these are divided by 16. So 1 would be this. Your probability of being little t, big S, would again be 1/16. Again, right there. Your probability of being big T, big S, little s. That's 1/2 times 1/2. So that would be 1/4, or 4/16. That's what we see right there. So this assumes true independence. Now, independence works well with this independent assortment thing. And we can see how this happens by looking at the chromosomes. Imagine here, let's say all the blues came from dad and all the reds came from mom. And this is now in the individual. This is chromosomes 1, 2, 3, and 4. Okay? You have your meiotic divisions, and then you produce this set of gametes. You can see along here, you may pass on the red chromosome 1, you may pass on the blue chromosome 3, you may pass on the red chromosome 4, etc. All sorts of different combinations happen. But in reality, when you're looking at genes along a chromosome, it's not that simple. Often homologous chromosomes, these chromosomes that basically came from dad and came from mom, will trade pieces during this process of meiosis. That was what was depicted in that diagram from Morgan. So this happens when they're doubled, and this process is called crossing over. And this again forms recombinant chromosomes, or recombinant gametes, where if you're looking at two genes, you can get combinations not found in the parents among the alleles. So let me just walk through this a little bit more slowly. So you have two homologous chromosomes, one inherited from the egg, one inherited from the sperm. So here's your egg chromosome, here's your sperm chromosome. You see them both. So here they are, they're put together. Okay. So what happens then in the individual, so let's say, for example, this is me, and the red I got from my mom, the blue I got from my dad. What may happen inside me in the process of making some of my gametes, I may have something like this happen, where pieces get broken and stuck together, such that among my eggs, I obviously didn't produce eggs, but among my eggs, I might have some non-recombinant, so this is something that passing on exactly what I got from mom, and some that are recombinant, which have bits from mom, bits from dad, bits from mom, bits from dad. This is a recombination event happening along a chromosome. So you may be wondering, how do we tell if recombination has happened? How can we tell that this is going on? Well, what happens is we look at genetic markers. And we'll talk about what genetic markers are, but you can think of them as sort of the big A, little a, and big B, little b that we've talked about in the past. You could think of them like the traits they're associated with, which thumb's on top, or being colorblind or not. And in this case, let's say, for example, we know that from your mom, you got the capital A and capital B. We know from your dad, you got the little a and little b, right? The non-recombinant eggs from this individual would be capital A, capital B, and little a, little b. And something that was identical to the sperm or egg that made you. The recombinant eggs would be those new combinations, little a, big B, and big A, little b. Okay? Now, importantly, you won't always have recombination. In fact, if let's say, for example, the A and B genes were right next to each other, let's say they're basically stuck together, at that point, you refer to them as being totally linked. They're linked together, that if you get the big A, you're always going to get the big B. If you get the little a, you'll always get the little b. If that was true, then you would only get these non-recombinant eggs, right? If basically these two things were glued together, then you would only get non-recombinant eggs. And what tends to happen is neighboring gene variants or alleles tend to stay associated. So I've diagrammed here what you might imagine looking at several chromosomes from one heterozygous individual. So again, here's the chromosome from mom on top, that's red. The bottom chromosome is from dad, that's blue on the bottom. They are producing gametes. You might see this along the gametes there. So you have a stretch of red, stretch of blue, stretch of red, stretch of blue, stretch of blue, stretch of red, etc., etc. If you look at this, how often are A and B linked? How often do you inherit a red A with a red B or a blue A with a blue B? A and B are right next to each other, and if you look across this whole chart, pretty much always. If you get a red A, you're always getting a red B. If you're getting a blue A, you're always getting a blue B. You see that very clearly along here. In contrast, how often are A and C linked? Notice A and C are further apart. Well, here in this first individual, A and C are both red. In the second, we actually get a red with a blue. Third, we get a blue with a red. Fourth, we get a blue with a blue. So really only about half the time are you getting the two parental types, and half the time you're getting a recombinant type. Now, this is what's used for constructing genetic maps. That if you have genes close together, they tend to be inherited together. You're violating this principle of independent assortment that I described before. Though what's happening in this case is you're looking along one chromosome rather than looking at different chromosomes. But you can no longer multiply the probabilities like we did before. So let's try to follow this. Now let's assume total linkage. Let's assume that A and B are stuck together. They're right next to each other. So let's imagine dad is big A, big B. Let's imagine mom is big A, big B, little b. So dad is always giving the same gametes no matter what. Dad is always giving a big A, big B, right? So we see that along this chromosome. Dad is always giving a big A, big B. Mom, in this case, will only give a big A, little b, right? Or a little a, big B, because we said it's linked, right? So this is what mom's going to give. So what kind of offspring can we see? Well, we can have big A, big B, little b, the same over here, or we can have big A, little a, big B, big B. So in this case, there's only two possible forms that you can get out of this. There's only two possible offspring if you have total linkage in this case, right? What would happen if, in contrast, there was no linkage? What would happen then? Well, again, dad is always going to be giving big A, big B, right? Because that's all he can ever give. Even if he did have a recombination event, we would never know because it would always still be big A, big B. But in this case, mom has more things she can give. Here we have the parental types, big A, little b, and little a, big B, right? But if there was no linkage, sometimes little a and little b can go together, or big A and big B can go together. So these are recombinants, and these are parentals. And as a result, there's a lot more possible options that you can get out. So we can get, in this case, big A, big B, little b, big A, little a, big B, big B, big A, big B, big B, and big A, little a, big B, little b. So in this case, now we have 4 possible types of offspring rather than just 2. Now, one thing I hope you noticed in looking at this is that phase was important, that basically which allele is next to the other one. In this case, let's say, for example, you were to say an individual is big A, big B, little b. You don't actually know the phase. You don't know what combination of alleles he got from the sperm that made him as opposed to from the egg that made him. In this top example, the egg that made him was big A, big B, and the sperm that made him was little a, little b. So that's one possibility. The other possibility is the egg that made him was big A, little b, and the sperm that made him was little a, big B. In each of these cases, you get different sorts of recombinant gametes. These bottom ones, look at the bottom set, the capital letter ones are recombinant gametes here, right? Because you have to have a recombination event to get those. For the top one, the one capital with one lowercase is what's recombinant. So it's very important that just because you see an individual as big A, big B, little b, you can't actually just from that alone figure out what sort of recombination is going on. So we'll pick up on this in the next video to look at how to calculate recombination distances between genes. Thank you very much. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the principle of crossing over. What we'll do in this video is leverage that principle to see how we can calculate recombination distances between genes. And later you'll see how we'll actually use that to construct a map of the genome, or a map of multiple genes at once. Let's first recap some of the points from the last video, just to remind you. Again, we can use linkage to develop a map of chromosomes. This is our ultimate goal. Now, importantly, as we said before, chromosomes are typically linear. Neighboring gene variants tend to remain associated even when you have crossing over. So if you have two genes that are very close together, you'll tend to inherit alleles of those genes together. Whereas genes that are further apart, you may be more likely to get recombinant forms from them. If they are very close together, you typically only get the parental types. If they're further apart, you may get some parental, some recombinant. So what you tend to see from this is the full range of possible associations. You may have no recombination or no crossing over, particularly in this case, between two genes. This would mean they're very, very close together. There's nothing breaking them apart. Or you could have no association, basically 50% recombination. This is what you see when things are very far apart on a single chromosome, or you tend to have them on different chromosomes, one of those two things. At that point, they are independent of each other if you get 50% recombinants, 50% parental. Now, let's start by looking at how we identify what's going on along these chromosomes. So I was depicting these chromosomes before and referring to points A, B, and C along them. But how do we know what variant is at A, what variant is at B, or what variant is at C? Well, the answer is we use genetic markers. The genetic markers are basically reference points in the genome that have two alleles or more. They have two forms or more. Now, you don't necessarily know where this is. Now, this sounds a little bit funny because you can actually generate a map without knowing where at all you are. You could think of it in the context of saying you know where something is relative to something else, but you may not know exactly where that is. So, for example, I could tell you there are three genes, ADH, ACT, and ALDH. And you can say that I know that ALDH is in the middle of the other two, but you don't know what chromosome any of them are on. That's okay. That's the start of how you put together a genetic map. Now, genetic markers can be molecular, right? Being molecular, meaning that you assay them using some sort of molecular biology technique, such as, for example, doing PCR, or the polymerase chain reaction, and assaying it on a gel. This depicts right here, the bottom left picture, different alleles. So this could be a big A individual, so that's two copies of the big A allele. This is a little a individual, that's two copies of little a. And here is your heterozygote, has one of each. Right? So this is a very easy assay there. In this particular case, what's happening is they run differently on the gel, that the smaller fragments migrate faster than the larger fragments. And there's a difference in size, where the little a has this extra set of bases in it that's not present in the big A. And that makes the little a run slower down the gel and tend to be higher up. That's just one example. Don't worry about the details of this, but this just shows you what we mean, or how people will assay a genetic marker. Now, genetic markers can be molecular, but they don't have to be. They can also be phenotypic. They can also affect the appearance of an organism. So we talked already about these cases of having red versus white eyes, these Drosophila mutations. Now, you can say, for example, this red-eyed individual may be big A, or big A, little a, because we know that the red-eyed one is dominant. This white-eyed female may be little a, little a. Now, there's obviously some underlying nucleotide difference, some underlying DNA sequence difference at an eye color gene that's making it so the little a individuals have white eyes. But we can nonetheless use these as a genetic marker. And in fact, this is something that was used extensively over the course of the last century, and still at some level today is used for making genetic maps, these sorts of phenotypes. This is also how we map genetic diseases, and we'll come back to that a little bit later on. Now, what we do is we use either controlled crosses or pedigrees. They're basically the same thing. Controlled crosses being when you take the red-eyed fly and mate to the white-eyed fly, or pedigrees is you're looking at things that have already happened and just inferring what's going on. So you can say that we know your great-grandfather had brown eyes, but we know your great-grandmother had blue eyes, and look at what's happened. And we infer the genotypes to make a map. Right? We infer the genotypes to make a map. So let's use the case of what's referred to as a test cross. Okay? So what we want to do is we want to identify parentals and recombinants. So we'll start with something which is homozygous, which has only one type. Big A, big B, little b, crossed to little a, little b, little b. Okay, so let's say the red is the female and the blue is the male. We make this offspring, right? So this is a heterozygote, big A, big B, little b. We know this is the phase. We know that you got the top one there from mom. got the bottom one there from Dad. What we do is we cross this to the homozygote again. So cross it to a little a, little a, little b, little b. Now, if A and B as genes are totally linked, what fractions should we get from all these different things down here? If there's no crossing over between A and B, what should we see? Look at that for just a second. If there's no crossing over between A and B, we'll only get the parental types. So in this case, the parental types will be big A, big B, this set, all right? And little a, little b, which would be this set. And then this individual always gives a, little b. These would not be found at all. If they were totally linked, you wouldn't get any of those. Okay, now let's do a different test. Let's say they're completely unlinked. Let's say they were even on different chromosomes. What would happen then? If they're on different chromosomes, you get all 4 types. And in fact, you would get 1/4 of each of these. So these are the parentals, these are the recombinants. Half of the overall offspring would be recombinants, because these are your recombinant set there. Now, the question that I want you to think about is what happens if, somewhere between these two extremes, not the totally linked and not the totally unlinked, like being on different chromosomes. What would happen if they're sort of linked, like they're near each other but not right on top of each other? Well, what ended up happening in that situation is you get mostly these, but you get some of these. Basically, this would be more than 50%, but less than 100%, and this would be something less than 50%, but more than 0%. What this recombination fraction tells you, what the fraction recombinant tells you, is how far apart the A and B genes are. The further they are apart, the more the percent recombination will approach 50%. The closer they are together, the more the percent recombination will approach 0%. So let's look at this using this particular fly cross. Now, here what we'll be doing is we'll be crossing flies that are mutant for one gene to flies that are mutant for another gene. So let's start with over here with these ones on the left. These ones have cinnabar or bright red eyes. So this is different from the standard fly eyes, which are more of a sort of a dull red. And let's say we're crossing males, let's put your boy symbol, to females that are vestigial. Okay? Vestigial has these sort of shriveled up wings rather than the normal, full-length, beautiful Drosophila wing we see. Now these boys here have normal wings. So we can say that they are cinnabar, so they have the CN, but they are VG+, because they have the normal copy, not the mutant copy of this particular gene. In contrast, these vestigial winged females, we can say they are CN+, because they have normal dull red eyes. They don't have the mutant bright red eyes. So we're crossing CN VG+ males to CN+ VG females. Okay? So these ones here are mutant for cinnabar. You're mutant for the thing that doesn't have the plus. So we're crossing these mutant red-eyed males to these mutant squished up winged females. Okay? Now, we cross these and the offspring all have normal eyes and normal wings. So what can we infer from this? Well, we can actually infer quite a bit. First is the cinnabar form, the bright red eye form. Is that mutation dominant or recessive? Right? Well, in this case, we know the mutation has to be recessive, right? Because we crossed a fly that was cinnabar to a fly with normal eyes, and in the end, the flies all had normal eyes. So normal must be dominant, or cinnabar must be recessive. Similarly, vestigial must also be recessive, because again, we crossed a vestigial winged fly to a normal winged fly, and the offspring all had normal wings. So normal wings is dominant, or vestigial form is recessive. So what would be the genotype of this fly? Remember we said that this one over here was starting off with CN VG+, this one was CN plus VG. Well, the genotype for this fly, when we're looking at basically the combination of alleles it has, would be something like this, where it has the CN VG+ from one parent, and it has the CN plus VG from the other parent. This is the combination. Now, we're going to assume that these are actually on the same chromosome, which actually they are on the same chromosome, but let's just assume that we're guessing that. Now let's say we cross F1 females, so these are what's referred to as the F1 females, okay, these offspring up here. Let's say we cross them to males that have the cinnabar mutation and the vestigial mutation. Well, what would the genotype be for these males? If they have both mutations, we know it's recessive, what would the genotype for them be? Well, since the males were exhibiting both mutations, the male is necessarily homozygous for CN and VG because it has cinnabar eyes and vestigial wings. Okay? So we're looking at, these are the males that we're going to cross them to. These are the F1 females right here. What would be the parental and the recombinant offspring? What would they look like? Well, let's think about this. This is always giving the mutant forms, right? So the dad's always giving the mutant forms. So basically from dad, you're always getting CNVG, no matter what. You're always getting that combination. It doesn't matter if there's a recombination or not, which there isn't any recombination in Drosophila males, but let's just leave that alone. If you're getting parental gametes from the female, what would you get? You would get either CNVG plus, or you would get CN plus VG. So what would these flies look like? Well, this top set of flies here would have the vestigial wings, so they would look like this parent. This bottom set of flies would have cinnabar eyes, so they look like this parent. But that would be it. You would not have any normal-looking flies, and you would not have any flies that look like their dad, that are mutant both. Recombinant offspring would get something like this. They would get the CN and VG from mom, which they'd also get a CN and VG from dad, meaning they would be mutant at both genes. Or they would get CN plus and VG plus from mom, meaning they would look totally normal. They would not have any mutant forms. So let's look at these. Here are all four possibilities. These top two depict what would happen in the context of just getting parental types. The bottom two depict the recombinant forms. So what if there was total linkage? What if the CN and VG genes were right next to each other? What if there's total linkage? What would you get? If there's total linkage, you'd only get these two forms, right? If CN and VG were completely unlinked, you would get half these forms and half these forms. So maybe a quarter of each of the lines there. But what if they are linked but not totally so? That's when you get mostly these and a little bit of these. Okay? Now, again, it's this fraction recombinant that reflects the distance between genes. That if genes are very close together, then the fraction recombinant will be low. If the genes are very far apart, the fraction recombinant will be high. Now, Thomas Hunt Morgan, who I mentioned, the Drosophila geneticist, he suggested this percent recombinant can be called MAP units. And often now in Drosophila, they're referred to as centimorgans in honor of him. So again, this recombination fraction ranges from 0% to 50% recombinant. And this is often just referred to as 0 to 50 centimorgans. Okay? Now this gives you an idea of the distance between genes. And this was known like 100 years before we had the first genome sequence. Not quite, but close to it. And at this point, we had a way of putting together a map and saying, this gene is close to this gene, this other gene is really far away. And the genes in this case were things like these Drosophila mutations, like the cinnabar gene, the vestigial gene, the white eye color gene, things like that. In humans, just for your reference, 1.3% recombination is on average about 1 million bases. That's not exactly the case, but that gives you sort of a ballpark that you can work with. And this is used to determine the linear order of genes. And we'll come back to how that's done in the next video. But let's put in some numbers here. Let's say that you were counting flies. So this top fly here has normal eyes but vestigial wings. The next one has cinnabar eyes but normal wings. The next one is normal at both, and the bottom one is mutant at both. Now in this case, they all have the mutant copy from their dad. This is that test cross. So they're all over CNVG. Because they are diploid, they do have 2 copies of everything. But what I drew it on here was just to depict the phenotype or how they look. Let's say you got these numbers. What does this tell you? Well, these are all the parental types, right? These are the ones that match. Basically, they got this gamete from their mom, or they got this gamete from their mom. So these are parentals. These lower ones here are recombinant because they had to get either this form or that form from mom. So these are the recombinants, these are the parentals. Okay? Now what fraction overall here are recombinant? Well, the total number of flies here is 200. The total number of recombinant here is 20. So the recombination fraction is 10%. There's 10% recombination between these two things. So this gives you some idea of distance between these two genes. Now, in the next video, we'll show how you leverage this to generate a map, and particularly using 3 or more genes and identifying the linear order of these genes. In the meantime, look online for some sample problems that you can try on your own. Thank you.